Welcome back, CAVE students. I'm Joanna George Johnson, and I will be your teacher for this biology lesson. Let's get into it. Today, we'll be looking at the different parts of the aerobic respiration, which is called the link reaction, Krebs cycle, the electron transport chain, and then we will talk about some important factors that deals with aerobic respiration as we go into it. So let's begin. So please remember that the link reaction and Krebs cycle occurs only when oxygen is present. And so we are going to start with the link reaction since it happens before. Both of these processes occurs in the matrix of the mitochondria. The mitochondria contains three major sections. The innermost section of the mitochondria is where we call the matrix. It contains all of the necessary enzymes, the fluids, the proteins, and so on that is required for the rest of respiration. And so therefore you find the link reaction and Krebs cycle will be going on in that section of the mitochondria. So what is the link reaction? Now, if oxygen is available, the pyruvate that is made at the end of glycolysis will move to the matrix of the mitochondria. Please be reminded that glycolysis occurs in the cytoplasm of the cell. So therefore, there is a movement of the pyruvate into the mitochondria for the further processing of the three carbons or the carbons that are coming from the original glucose molecule. Now, you will see in some books that instead of seeing pyruvate, you'll see pyruvic acid. It's basically the same thing. Those of you who do chemistry would know that when you have an acid, even an organic acid in water, what happens is that there's a hydrogen ion that dissociates from the acid, and the resulting part, you have the anion and the hydrogen ion. That is how it shows up in mixed into water. And because all metabolic reaction takes place in water in a cell, then typically pyruvic acid will actually shows up as pyruvate. Hence why I tend to use that form of the word. However, if you see in your book or books that you are reading, pyruvic acid is basically the same thing. Basically, during this process of the link reaction, pyruvate or pyruvic acid, as the case may be, is going to go from a 3-carbon compound to a 2-carbon group that will be attached to this substance called coenzyme A. So let's talk a little bit more about that. All right? Obviously, if you're going from a 3 to a 2 carbon, clearly there's 1 carbon being lost. And that carbon being lost will come off as CO2. All right? Please bear that in mind. So let's look at it. First thing is that the pyruvate will lose one of its 3 carbons during this process. And one of the a molecule of CO2 or carbon dioxide will be formed. So the process is known as decarboxylation. Why? Because, well, you're removing a carbon, decarboxylation. All right? Then, on top of that, 2 hydrogens are going to be removed from the pyruvate as well. And this is because you're removing hydrogen. It's called dehydrogenation. But in a strict sense, what happens is that these hydrogens are going to be picked up or captured by the oxidized form of NAD+. And then when it's attached, it is called reduced NAD. Remember from previous classes I said to you that anytime a compound or a molecule gets hydrogen attached to it, it is being reduced. When it loses the hydrogen, it is being oxidized. When it gains hydrogen, it is being reduced. All right? That's just a chemical or chemistry term for the process. Now, the 2-carbon molecule that is now made, called acetyl group, it will combine with a substance called coenzyme A. And it is combining with coenzyme A to make acetyl coenzyme A. Yeah, nothing too fancy. Now, I'm going to show to you where this acetyl coenzyme A is very important to the whole process of not just respiration, but the fact that not only carbohydrates can be used for respiration. Typically, you would have heard that some, people say fats can be used and other substances. Now, here we're going to show you how fats or lipids can be used in the process of respiration. Clearly, fats do not enter glycolysis, but the making of acetyl coenzyme A is important for the next step of respiration, which is called Krebs cycle. And so if we go over here to this whiteboard, I have drawn on the board a diagrammatic view or representation of what's called a triglyceride. Okay, this part you should remember, these three chains, and I drew out one. The others I use the zigzag pattern that you should be familiar with from unit 1. What happens is that they can be each detached from the central part of the molecule called, as you know, glycerol. Okay, and these three things are called the fatty acid chain. Okay, now these come in very important because in a triglyceride, what happens is that it is possible, you remember the acetyl group? If I were to get an enzyme and you cut this fatty acid chain here, you cut it here, you cut it here, and so on, what you have here essentially is acetyl groups being made. And each of these acetyl groups, 2 carbon groups, can be now attached to coenzyme. Coenzyme A, making more acetyl coenzyme A. So just to let you know, the link reaction can occur with pyruvate and usually does, but then if there are times that are needed where your lipids in your body needs to be used for respiration purposes, this is where the lipids or your fats enters respiration, is at this stage. Just for the mere fact that the acetyl groups can be made just from cutting the fatty acid chains into 2 carbon groups. And if you notice, each of these groups can now enter Krebs cycle. And because there are 3 chains for each molecule, it means then, and each chain is quite long, it means that several of these will be made per triglyceride. Whereas in glucose, you only can make clearly only 2 of these per glucose molecule. So that's why you'd hear persons saying that fats provides more energy to the body because it has that ability. But just to let you know, it's not always the case. Okay? Let's move on. So, for each pyruvate molecule, the link reaction will produce the following things. 1 molecule of reduced NAD, and I put beside of it what the potential ATP is going to be. Please remember, the point of respiration is to produce ATP. So if you're not producing ATP, there's no point. So this is the reason here. The reduced NAD is going to later on go on to produce some ATP molecules. Each time you see it from glycolysis coming down, that is the purpose of it. Also, 1 molecule of CO2 will be removed as well. And so you can recall that there are 2 pyruvate per glucose because pyruvate is a 3 carbon compound and glucose was 6, which means it was split into half. So you have 2 3 carbon compounds of pyruvate. So each pyruvate is producing 1 NAD and 1 CO2. It means at the end of it all, the glucose molecule would have been producing 2 NAD or reduced NAD. I have 1. It's supposed to be 2 CO2 molecules and obviously 2 acetyl coenzyme A. And so far, you have broken 2 bonds, 2 carbon-carbon bonds from the original glucose molecule, which is very important because each time the carbon-carbon bond is broken, it means that there's a possibility for energy to be formed. Okay? Because just remember, the bonds were held together because of some energy force between them. Them being broken is going to release the possibility of ATP being formed, which is the energy currency of all cells. All right, so that's the link reaction for you. So after the link reaction, the acetyl coenzyme A goes on to the next stage of respiration. Okay, the next stage of respiration is called the Krebs cycle. It also occurs in the matrix of the, it also occurs in the matrix of the mitochondrion, just like the link reaction. What happens is that the acetyl coenzyme A attaches to a 4-carbon substance called oxaloacetic acid. I wish I was able to write it out here, but I didn't. But just notice that this is a 4-carbon compound here. Remember, the acetyl group is a 2-carbon compound. So when this 2 adds with this 4, it's going to make a 6-carbon compound that is here called citric acid. And some of you might be familiar with this particular acid. This is the same acid you find in your oranges, your limes, your lemons, and grapefruit and things like that. It's the same acid. All right? And it's a 6-carbon acid. But please bear in mind, what I usually do is that I color the 2 of the carbons in the group just so you can remember that these 2 carbons are coming from the original glucose molecule. So as the acetyl group attaches to oxaloacetic acid, what happens is the coenzyme A, which is just like a taxi, it just brings it to the position to be attached to acetyl coenzyme A. After that has been done, coenzyme A goes off to find the next acetyl group. All right? Now, the cycle begins after that. What happens is a series of complex enzyme-controlled reactions obviously will happen. And please note, some of the different levels, you're not needed to know all of the names of the different parts. So it's just make it easier for you just to watch the number of carbons as it goes. So first thing to happen is in the process that there is decarboxylation that is taking place. So one carbon is lost as carbon dioxide. So far we have lost 1 carbon dioxide up in the link reaction, and next one here, right at the start of the Krebs cycle. And then on top of that, you're also making more reduced NAD. All right? And reduced NAD, all right, as you know, will go on to form ATP later on in the process. Now you move from a 6-carbon compound here to a 5-carbon compound here. From 6 to 5, because you have lost one of the carbons. So one of the carbon that you have lost, we're going to call that's a carbon that originally came from the glucose molecule, and there's only one now left. I'm just marking it as much as I can here. So what happens is, as the process continues, a next carbon dioxide comes off. So here you have a next carbon being lost. I'm just marking it up so you can recognize. So this is the second carbon that we started out with at the start that is now being lost. So you go back to our next 4-carbon compound. Just to let you know, you don't go back to oxaloacetic acid immediately. There's a series of reactions that takes place before it goes back to oxaloacetic acid. The point is that during this process, NAD, reduced NAD is also made again. And then, as it goes from one compound, 4-carbon compound, to a next 4-carbon compound, actual ATP is directly made. And we'll talk a little bit more about that. And then, instead of NAD reduced NAD being made, this time you have reduced FAD being made. Now, FAD is similar to that of the NAD. It will do the same job, but it's not as efficient as reduced NAD. But this is what actually happens during the whole process of Krebs cycle. Anyway, on its way back to be converted into oxaloacetic acid again, a next molecule of reduced NAD is made. Okay? Now, class, I want you to bear this in mind. So far, we have lost both of the carbons that we entered Krebs cycle with. So by the end of Krebs cycle, all of the glucose carbon-carbon bonds have been broken. Because, and also please remember, because if you're starting with glucose, you would have had 2 groups of pyruvic acid starting out with, which means there were 2 groups of acetyl coenzyme A, which means that glucose will turn Krebs cycle twice. So all of this process is going to happen 2 times for every glucose molecule we start out with in glycolysis. So these 2 carbons will be lost. So by the end of this cycle, the first time around, 2 carbons will be lost. The second time around, and next 2 carbons will be lost. So if you add them up, this 2 added to the next 2 that will be lost from the second spin of the cycle, that's 4 carbons in total. And remember, during the link reaction, there was a carbon that was lost again per pyruvate, which means now you have all of your 6, because 2 from up here, because this is going to happen twice per glucose molecule, and then 2 from here, that is going to be times 2, because this is going to happen twice. Okay, sorry for the bad handwriting, but so it goes sometimes. Okay, I hope you are getting this, because it's very crucial that you understand what is going on. Now, before I go any further, I need to talk to you a little bit about the making of the ATP molecule. Now, the direct making of the ATP molecule is just that in the conversion of this compound to this other compound, it's still a 4-carbon compound, so there's no loss of carbons or anything like that. But that conversion is enough to allow for ADP to attach to what's called an inorganic phosphate. That inorganic phosphate is just because of in the matrix of the mitochondria, there are a lot of different ions and stuff floating around in there, and therefore you have ADP attaching to the inorganic phosphate to make ATP. Remember, the T in ATP stands for 3 or tri. So that's why you have D, which is 2, add 1, make 3. Okay, very simple. And so this form of phosphorylation, from photosynthesis we told you already, that phosphorylation is about the addition of phosphate to something. And so in this case, the making of ATP is always called phosphorylation. The making of ATP in photosynthesis was called photophosphorylation. Photosynthesis. The making of ATP in glycolysis, because it's used the same method, is called substrate level phosphorylation. And the making of ATP here in Krebs cycle is also called substrate level phosphorylation. Anytime the ATP is being made directly, that is how we end up with substrate level phosphorylation. The reason I'm stopping to say this to you is the next step we're going to look at is a different type of phosphorylation that is going to occur. All right? I'm just going around. This is still showcasing all of Krebs cycle. And Krebs cycle, all what I just said in words. So I hope you're able to see this and recognize what I just said is what happened from the acetyl coenzyme A leaving off the acetyl group to combine with oxaloacetate or oxaloacetic acid to make the 6-carbon citrate. When this happens, the coenzyme A goes to its own way to pick up a next acetyl group, and the cycle continues. At the end of this whole process, Krebs cycle, what happens here is that 3 molecules of the reduced NAD will be made per pyruvate. So clearly, if it's 3 molecules, you have to always multiply this by 2 if it's pyruvate, because we are looking at the entire glucose molecule. So the entire glucose molecule would have done this 6 times instead. All right? Each pyruvate will have produced 1 molecule of reduced FAD, 1 molecule of ATP, and 2 molecules of CO2. Okay? So, and recall again, always remember that the 2 pyruvates would have been made from 1 glucose molecule. So the numbers you're seeing above should be doubled for the full tally, which we have here. So by the end of Krebs cycle, you have made all of these NADH and carbon dioxide and FADH. And please note, by then, there's no more carbons from the original glucose molecule to contend with. So now we are going to focus on making more ATP, but this time, we are going to be looking at all of those reduced NAD and reduced FAD that were made in the previous stages. So now we are going to go to the electron transport chain. And it's sometimes called oxidative phosphorylation. Okay? Oxidative phosphorylation, because this chain would not occur if oxygen was not available. All right? And please note, the link reaction, Krebs cycle, all of those would not have happened if it was not for oxygen being present in the cell. All right? Now, this diagram can be a bit intimidating, because a lot of students see it and they go, oh Lord, what is this? So let us try and see if we can demystify it a bit. It's a chain. These circles, these blue circles, actually represent the different protein complexes that it will be accepting the hydrogen, the electron, from the reduced NAD, thus oxidizing it eventually. And also, please note that you notice these gray section called the inner membrane here. The gray part represents the double membrane, or the lipid bilayer, because you should remember that from Unit 1, that a membrane is made of a lipid bilayer that is going to allow for the transfer of substances, all right, because it's a selectively permeable membrane. Now, what happens at the start of the chain? The reduced NAD will first dissociate the hydrogen from its electrons. Well, not all of its electrons, obviously. An electron, which will be captured by the first molecule in the protein complex, the chain of protein complexes. You don't have to know the names of them. So that's okay, all right, that you don't know. Just to know that just like with photosynthesis, the electrons are going to be transferred from one complex to a next, to a next, to a next, to a next, to reach the end of the chain. While this is happening, the hydrogens that are coming off from the reduced NAD are going to make their way through the membrane to the other side of the membrane. Now, this process is actually a sort of active transport, but this is an active transport that don't use ATP. But, miss, how can that be? I thought we were told that active transport requires ATP. Well, I'm not certain who told you that, but what you should have been taught was that active transport requires energy to move substances from one side of a membrane to the next side of the membrane. And in this case, ATP is not the energy that is being used. What is happening is that as the electrons is transferred from complex to complex to complex along the chain, there is enough energy given off during that transfer to allow for the movement of the hydrogen ions through the membrane to the other side. Okay, it's very important. Now, at the end of the chain, the final acceptor of the electron is going to be oxygen. And also, the hydrogen ions will come into play, and water will be made. So if you remember correctly from the equation of respiration, that at the end of respiration, you had 6 carbon dioxide being made per glucose molecule and 6 molecules of water. Here is where the water is being made. Oxygen is a final electron acceptor, and hydrogen comes along, the hydrogen ions that is, comes along to bind with the oxygen to make water. Then you're going to say, but miss, the hydrogen didn't go across to the other side of the membrane, so how will the hydrogen come from? Yeah, let's get to that. Now what happens is that the hydrogen start to get accumulated in this side of the membrane. And as they get accumulated here, they're now going to have a higher concentration up here than is down here. And you know what happens? They are going to want to come back to the other side, like in diffusion. But because they're charged, they can't just pass through the membrane just like that. So instead, they pass through a particular protein that is found in the membrane known as ATP synthase. And you would have seen that in the previous time when we did photosynthesis. It is the same molecule. As the hydrogen passes through the ATP synthase, it is allowed something to spin in the ATP synthase, which is a protein, it's an enzyme, and allow for the formation of ATP. Because this hydrogen is going to go and bind with the oxygen along with the electrons, it is called, this type of ATP formation is called oxidative phosphorylation, because oxygen is involved in the process somehow. And the process of having the hydrogen coming through the ATP synthase through the membrane is called chemoosmosis. Okay? So I hope you're understanding, and I've drawn this part to show this is outer membrane of the mitochondria. This is the inner membrane space. This is the actual membrane or the cristae, and the matrix in the center. All right? So this is the end of respiration, where at the end of respiration, what you have happening is that the reduced NAD is now being converted through a process so that ATP can be made using ATP synthase. All right? So at the end, 33 ATPs would have been made based on all of the NADH and all of the FAD. Oh, I did not show that to you. It was in the diagram where FAD actually joins the chain later on, not at the start of the chain. So that's the reason FAD is usually accredited for less ATP formation in comparison to the reduced NAD. All right? Well, we have heard of a cyanide. We have heard of cyanide. And cyanide is a poison. Everybody knows about cyanide, how cyanide is a poison. Now, how is cyanide poisonous? Here is why. This is a question that comes about the poison ability of cyanide, because cyanide is what's called, it has aerobic respiration. How does it do it? What happens is, at the end of the electron transport chain, instead of oxygen being the final electron acceptor, cyanide ends up being the final electron acceptor. And if that occurs, it means there's no making of FAD anymore. Oh, sorry, there's no making of the oxidized FAD plus or NAD plus. So what ends up happening is that it halts respiration altogether, because at the end of that chain, the right thing is not there to accept the electron. So cyanide has basically stopped respiration right at the end, and therefore it backs up everything else, including Krebs cycle, because Krebs cycle requires FAD plus and NAD plus for it to make reduced FAD plus, FAD, and reduce NAD. And if they are not available, then Krebs cycle cannot go either. So we have to watch that. Please note that human skeletal muscles cannot respire anaerobically forever and ever. Amen. Okay? Also note that your heart, I have to go through this very quickly, but your heart muscles cannot undergo anaerobic respiration any at all, because anaerobic respiration will cause a muscle to get tired, to get weary. Could you imagine if your heart muscles started to get tired? What a thing that would be. If your heart beating from before you were born, the muscles say, you know what? Because there's no oxygen around, we're going to respire anaerobically. If that was to happen, it would mean then the muscles of the heart would start getting tired. We cannot afford for our muscles in our heart to get tired, because the lactic acid buildup during anaerobic respiration is toxic. You can't afford for toxins to be in your heart. That's one. Two, the body, when the heart gets tired, and when any muscles get tired, eventually it will stop or slow down. We cannot afford that to happen with our muscles in our heart. And so that's the reason, even though you may have heard that anaerobic respiration can take place in humans, it only takes place in the skeletal muscles, your leg muscles, the muscles in your arm, and so on. They can afford to get tired, because when you get tired, you sit down, you cool out, and you rest. Okay? We know that when we get tired after a long bout of exercise, one of the things that happen is that fatigue brings some pain to the muscles. And those sore muscles will make you not want to move again, not want to do that exercise, even though you should, like myself. But guess what? If your heart muscles were like that, then we'd have a problem, because your heart can't afford to be in pain while it's doing its job. So because the heart has to be constantly working, even when you are sleeping. You understand? And so because of that, anaerobic respiration will cause fatigue, as we told you before. The heart cannot afford to get tired. And so therefore you find anaerobic respiration cannot take place in your heart muscles. And next place anaerobic respiration doesn't really take place is in your brain. Okay? Because your brain cells cannot afford to stop working at any time because it's tired. Because you need your brain even while you are sleeping. Okay? So I hope we have done a good job so far with telling you about respiration. If you have more questions, and I am going to post this in the Google Classroom. I know I didn't post one last week. It's because it's a continuation of this one. So therefore, I'll post this whole thing, and so you have access to it, because I know there's quite a number of people that is already there and who are waiting for it and have been asking for it. So I'll do that for you, okay? So the main points of this lesson for you to remember, class, is this. Respiration is the key way of cells getting energy. This starts out from releasing that energy from organic food, such as carbohydrates.